geneXplain speaking here. Today our Chief Scientific Officer, Alexander Kel, to tell us something about a new tool called Genome Enhancer. So, Alexander, what is Genome Enhancer?
Okay, so let's start from our knowledge. So we know that our DNA, our genome, contains gray zones. So those regions of genome which are actually occupy most of the genome, more than 80%, which are not coding any genes, but they contain inside so-called enhancers. And those enhancers are usually sleeping. But in many other diseases, especially in cancer, those enhancers for some reasons are waking up and they start controlling genes in the wrong way, and finally genes start doing wrong things. So we developed a new tool which is called Genome Enhancer, which is able to identify those enhancers wrong enhancers in disease genomes, and through that to help us to understand the molecular mechanism of disease.
Okay, so and what types of data do you need to run Genome Enhancer?
So the Genome Enhancer takes so-called omics data. So using special new technologies of sequencing, we can analyze samples of molecular samples and actually reveal all genes which are expressed in these tissues, so-called transcriptomics. We also can identify all mutations in genome using also sequencing and so-called genomic studies or omics exome studies. We also can identify the changes in methylation of DNA, so-called methylome. So those omics data are input for our Genome Enhancer tool.
And what does Genome Enhancer do with this data? What is the analysis?
So the Genome Enhancer takes all this omics data and is doing so-called multi-omics analysis. So it checks whether different omics data help each other to analyze first of all, what changes in genome are happening in disease tissue, whether those changes are influencing on the corresponding genes and changing the expression of those genes, or this is maybe a result of modification of DNA through methylation of other changes in genome, which lead to those waking up of those enhancers. So with this multi-omics analysis, the Genome Enhancer actually, after revealing these enhancers, it identifies so-called master regulators in the disease. So those elements of signal transduction pathways which are acting through those enhancers to wake them up. And once we identify those master regulators, the Genome Enhancer tool can look at the database and identify actually drugs which can influence on those master regulators, on those targets, and propose those drugs as a potential therapeutics against the disease we study.
So, starting from raw omics data, the analysis goes to the master regulator identification, and then some suggestions for therapeutic agents to
Exactly. That's
okay. And who is this program for? Who should use Genome Enhancer?
Actually, we developed this tool for many users, and we also made such a very easy interface. So basically, once you have your omics data done and you have your data files in your computer, you simply drag and drop them in the special box on the website, and that is a very nice graphical interface. You specify which files belong to your disease tissue, which files belong to your control, and then what happens, you press one button, which is called run, and then you go to drink your coffee. After a couple of hours, you come back, and this tool is actually generating a full report of all the analysis which was done. And the interesting thing, it's actually generating it in the form of a scientific paper.
Wow.
So it discovers the drug targets and put them in an abstract of that paper, and then it describes what was done, what data was taken, what result was appeared to be found, And it's also generate all the pictures, tables, and at the end it's also made a little summary about our discovery. And actually it's also described very well what kind of algorithms were used. So then when you want to publish your study, you know exactly what was done with those tools. So all in all, it's a full report which is ready to understand what's going on and ready to submit as a scientific paper.
Wow, just incredible. So, who is the end user of Genome Enhancer, taking into account everything which you have just said?
Yes, so basically it's any researcher who studies omics data in their samples. It could be a molecular researcher who is analyzing the basic mechanism of disease using their cell lines. It also can be a medical researcher who has a collection of their samples from different patients, and he wants to summarize the analysis and understand the mechanism of the disease in those patients. It can be also a doctor who studies a particular patient and basically takes the omics data from that one patient and he puts it in this Genome Enhancer, and then the program offers analysis of that personalized data, and it actually identifies those targets, those drug targets, which are specially activated in that particular patient, and also offers these therapies which may be specific for that particular patient.
So it's actually way beyond the state-of-art software for doctors where only the mutations are studied.
Exactly. So since we pay our attention to this gray zone of genome, so those places which are so far were not in the focus of many studies, so we can actually interpret those mutations, those majority of mutations which we observe in genome and which are happening not inside the body of the gene but between the genes. And then with that, we actually enhance our understanding of genome and enhance our predictions.
Wow, so I think I have only one last question left. How can we try to use Genome Enhancer? When will you supply it to us and where to get it from?
Just wait a little bit. So
okay.
we are almost ready to roll out this tool. So it's already ready. So it's in a beta version, it's already under testing. And in a couple of months, it will be there on the market, so you can try it.
Great, so we'll be waiting for that. Stay tuned to geneXplain. Thank you for watching. Bye.
Thank you very much.